lecture series with the jointly with the Austrian Academy of Sciences is therefore of central importance to us and to this end. Let me mention the previous speakers that we have welcomed here together in the series since it was inaugurated in 2016. David Gross, Christiane Nüsslein-Vollhardt, Martin Heira, Bernhard Schöllkopf. Last year, Angelika Amon, who sadly, after a very inspiring talk, passed away a few weeks after that talk. And this year, we are very, very honored to welcome the first chemist to speak at this series. And to introduce the speaker, I'll now hand over to Stefan Frauenberger, one of the first professors of chemistry at IST Austria. Stefan?
Yeah, it is a real honor for me to introduce to you Professor Carolyn Bertozzi. She's one of today's most prominent chemists and working at the interface of chemistry and biology, and particularly on glycoscience and the link with diseases. She's currently the NT and Robert M. Bass Professor of Chemistry at Stanford University, and at the same time investigate the Howard Hughes Medical Institute, which is a very prominent non-profit American medical research organization. Previously in her career, Carolyn Bertozzi has studied chemistry at Harvard with initially main interest in organic chemistry. Then she made sort of a switch to physical chemistry and electrochemistry at the Bell Labs. So I'm also an electrochemist, and from this, I know that in this time in the early 90s, she has proven to be also a very influential electrochemist. Then during her PhD in chemistry at UC Berkeley, she started to work on chemical synthesis of oligosaccharides, which was also the start of her current field in the research of glycobiology. And since 1996, she has been in the Faculty of Chemistry at UC Berkeley. And there she founded the field of Bio-Orthogonal Chemistry, and a term that she coined in the early 2000s. And this field goes back to her posing the question, is it possible to do organic chemistry reactions inside a living organism? So in answering this question, she has pioneered this field of bio-orthogonal chemistry, and her current research focuses on changes in cell surface glycosylation related to cancer, inflammation, and bacterial infection. Professor Bertozzi, as you can imagine, has received a very large number of awards, and by far cannot list all here, and I just want to name a very few. So already at the age of 33, she has been awarded the MacArthur Award, which is called also a Genius Award. She has been the first woman to receive the Lemelson MIT Prize, which is a major prize for inventions. Of course, I would say she's also a member of the National Academy of Sciences of the USA and also the German Academy of Sciences. Her fundamental research has not only pioneered her own research field, but she keeps finding many ways into applications. So Carolyn Bertozzi has also founded already a number of startup companies and keeps founding more. She's also a very inspiring mentor for young scientists, and a number of her alumni have become leading academics themselves. So before handing over to Professor Bertozzi, I want to mention to the audience, if you have any questions, please post them into the question and answer field, and so that they can be treated afterwards. And so with this, I hand over to you, Professor Bertozzi.
Thank you so much, Stefan. And let me just also start by thanking Dr. Zeilinger from the Austrian Academy of Sciences, Dr. Hensiger from the IST, as well as all of your colleagues who nominated me for this lecture. And it's just a wonderful privilege for me to have the opportunity to share some of the work from my lab with this distinguished group of scientists in the audience. Let me share my screen. So there we go. So as the title of the lecture would suggest, the focus of my lab is basically in the therapeutic opportunities of this fabulous field called glycoscience. And glycoscience means the study of sugar molecules. And it's an area of science that I've been interested in ever since my days as a graduate student at UC Berkeley in the late 1980s. And back then, the focus of my research was in the chemistry of glycans, how to synthesize them and put them together from their building blocks. And then I became interested in the biology of these molecules. And so that prompted my transition over to an immunology lab as a postdoctoral fellow. And then when I started my lab, originally at UC Berkeley, it was with the idea of combining the chemical tools that chemists love to create with the biological problems, many of which had to do with human health and human disease. And so today, the particular human disease that I'll talk about is cancer, and the work that we've been doing to develop a new class of immune therapies to treat cancer that are based on some discoveries we made from Glycoscience. So knowing that this is a very broad audience, and many of you are neither chemists nor biologists by definition, let me just start with a big picture overview of Glycoscience. It turns out that in addition to their function as nutrients, we all think of sugar as part of the foods that we eat that give us energy. There's an entirely different area of biology that these sugars engage in. And in us humans, that biology is very much the biology of the cell surface. So I like to analogize our cells to this very familiar candy, the peanut M&M. And we all know that when you bite through the middle of a peanut M&M, you'll see that there's this shell around the outside, which is the sugar coating. And then on the inside, there's the fats and the lipids, you know, the chemicals as a chemist would think of them, and the proteins and the nucleic acids and all those other biomolecules. But the surface of that cell is very much a sugar coating. Now, a century ago, when the sugar coating on the cells was first discovered using early primitive microscopy techniques, there was an assumption that that sugar coating served the same purpose as the sugar coating on the peanut M&M, which is a kind of protective shell. But then, as the century went on, it became clear that the sugars on the cell surface are far more complicated than that, and they have very important active biological functions. So now we think of the cell surface sugars more like the surface of the planet Earth. They are diverse in their features. Some of them are long structures like giant redwood trees in a forest. Others are short structures very close to the cell surface membrane. And if you fly over the surface of a cell, it might look something like this. And they're dynamic. These sugars are moving, and they're very actively contributing to the interactions of that cell with other cells and other components in the cell's environment. So nowadays, we think of cell surface sugars as a kind of a language. They're a biological code, and they can tell you something about the state of that cell. For example, those cell surface sugars is a language the cell can use to tell your immune system whether you're a healthy cell or a diseased cell. And that's how I got interested in the biology of glycans, or in Glycoscience. So in particular, more than half a century ago, it was recognized that when a healthy cell transforms into a cancer cell, there are changes in the cell surface sugars. And one of the most striking changes, which is observed in many different types of cancer, is that there's an abundance of glycans that terminate with a sugar called sialic acid. And so this class of glycans we call the cyaloglycans. And as this cartoon would indicate, a healthy cell has a pattern of cell surface glycans, and they're complex carbohydrates. So they're polymers, sometimes branched polymers, that are made from individual sugar building blocks. And the sialic acid is one of those building blocks. And it's usually at the very tip of the glycan, far away from the surface of that cell. And normal healthy cells have a certain pattern of these cyaloglycans. And that's important, actually. That's a signature that the cell is healthy, to have this healthy pattern. But cancer cells transform this well-manicured garden of cyaloglycans into this unruly tropical rainforest where they become much denser and richer. And it's really quite striking. And this has been known since the late 1950s. People found this when they analyzed cancer tissues that were removed from human patients by surgery. But until maybe 10 or 15 years ago, no one had a very good understanding as to why this might happen. Like, why is it that tumor cells, that these cancers, have this overproduction of cyaloglycans? So this is something that my lab started working on back when we were at UC Berkeley in the early 2000s. And what we discovered is that that overproduction of cyaloglycans is actually part of a program that cancer cells can use to undermine our immune system. So let's talk a little bit about how the immune system plays a role in protecting us from cancer. Your immune cells are your white blood cells, and they're coursing through your body as your blood is pumping, and they're continuously circulating out of the blood and into the tissues and sampling your cells to determine whether your cells are healthy or diseased. Of course, your immune system also is looking for foreign invaders. Your immune system, these cells are looking for viruses to get rid of, bacteria to get rid of, but they're also checking your own cells to determine whether they're okay or not okay. And they do this by forming a synapse with your cells. And they have a collection of protein receptors on their surface that are binding to ligands that are on the cell. And some of those ligands are going to be signatures of good health, and others are signatures of disease. And so the immune cells are just tasting every cell with all of these receptors and making a decision about whether that's a good guy or a bad guy. And if it's a bad guy, they will kill that cell and get rid of it. So let's talk a little bit more about that synapse between the immune cell and that diseased cell. The immune cell has receptors. These are proteins in the membrane. And some of those receptors are activating receptors. And if those kinds of receptors engage with a molecule on the surface of the target cell, and that molecule is an indicator of disease, and cancer cells actually present these indicators of disease because your body needs to get rid of them. So these activating receptors, when they cluster at this synapse between the immune cell and the cancer cell, they'll kick off a signal that activates the immune cell. And that immune cell will respond and say, oh, I've got to get rid of this cell. It's probably damaged or cancerous. Time to get rid of it. And there's different kinds of immune cells, and different immune cells will kill that target cell in different ways. But at the end of the day, enough of this signal through the activating receptor will lead to a reaction against that diseased cell. But it's a bit of a dangerous situation, because if that immune cell makes a mistake and accidentally reacts against a healthy cell, well, that will lead to an autoimmune disease. Your immune system will be attacking your healthy cells, and we don't want that. So there's this beautiful system built into our immune cells where, in addition to having these activating receptors to kill a target cell, they also have inhibitory receptors that just do the opposite. These inhibitory receptors are looking for signatures of good health. And if they find those signatures, they'll kick off a reaction inside the immune cell that literally shuts down the immune cell so that it will go away. So now think about cancer. Cancer is an evolutionary disease. It starts with mutations in the DNA that give the cell different properties. And some of those properties are signatures of damage, so your immune system can come and deal with the problem. But every once in a while, a cancer cell might figure out how to present these orange signals, these signatures of good health, and cloak itself with signatures that make it look like a good guy. And when it does that, it will trick the immune system into leaving it alone and deliver these inhibitory signals to the immune system. It's literally hypnotizing the immune system so that that cancer cell can survive and grow and grow and grow. So by the time a person has a tumor, like a big ball of billions of cancer cells in their body, chances are that tumor is there in part because those cells are tricking the immune system into leaving it alone. So this was a very important discovery in cancer immunology, and it led to what are now considered some of the most exciting new cancer medicines, which are the immune therapies. And in particular, there's an immune cell called the T cell, and the T cell has two inhibitory receptors called PD-1 and CTLA-4. And what was discovered now 20 years ago is that if you block these inhibitory receptors and prevent the cancer cell from hypnotizing the T cell, and you can block them with medicines, this can be a very effective way to treat certain cancers. And I'll just show for anyone in the audience who is interested in the technical jargon of immunology, a cancer cell can present a signature of damage, of disease, through molecules called MHC class 1 or 2. And there are peptides. This little black ball is a peptide in this presenter receptor. And that peptide has mutations in it that are a proxy for the DNA damage that this cell has suffered. And the T cell has an activating receptor called the T cell receptor, or the TCR. And when the T cell receptor engages this complex, it kicks off a reaction and the T cell will kill. But at the same time, if the cancer cell has figured out how to present a molecule called PDL1 or another molecule called B7, these molecules are signals of good health. And those signals will engage the so-called T cell checkpoint receptors, PD-1 and CTLA-4. And when these receptors get pulled together in the same region as the T cell receptor, they can shut down the activating signal. Biochemically, the way this happens is PD-1, for example, it has amino acid sequences in these yellow boxes, which have these acronyms, which are not important, but they're called the ITSM and the ITIM domains. And these regions can interact with enzymes called phosphatases, and they have the acronyms SHIP1 and SHIP2, the SHIP phosphatases. And those phosphatases will cut off the phosphates from activating signaling molecules coming through this positive channel. And that's how they shut down or quench the activating signal and turn off the T cell. And this discovery led to the development of a whole bunch of new medicines. And these medicines are monoclonal antibodies. They're big proteins, and these antibodies will bind either to PD-1 or to PDL1 or to CTLA-4 and just block the interactions that would quench the activating signal. And the big blockbusters among them so far are the Merck drug Keytruda, the Bristol-Myers Squibb drug Opdivo and Yervoy. Genentech and Roche have Tecentriq, and so on and so forth. And there's a whole bunch of these antibodies that are in clinical development, will eventually be approved drugs. And this was a discovery that led to the Nobel Prize in Physiology and Medicine back in 2018. Now, what does all this have to do with cell surface glycosylation? Well, it turns out that there is a family of sialic acid binding receptors that are expressed on all human immune cells. And this family, they're called the sialic acid binding immunoglobulin-like lectins, or the SIGLECs. And they are found on virtually every type of immune cell, and sometimes in combinations. And there's 14 of these receptors in humans. The reason we thought this was interesting is that those T cell checkpoint inhibitors, those blockbuster drugs, they can work to activate the immune system against cancers in some patients, but not in most patients. So some cancer patients, when they get immune therapies, they have a cure. It's stunning, it's remarkable. They go into permanent remission, but that's only about 20 or 30% of the patients. Most patients who get those immune therapies don't have that kind of a response. Their immune systems are not getting activated by those drugs. And the big question in this field is, why not? Is there some other mechanism that cancer cells are using to trick immune cells into leaving them alone? And we thought about this SIGLEC family of receptors, because if you look at their cytosolic domains, and so in this cartoon, the yellow line is the plasma membrane of the immune cell. What's outside the cell are the SIGLEC sialic acid binding domains. And there's some differences between the members of this family in terms of the size and the height, but they're all very similar. They bind cyaloglycans. But inside the cell, they have the same exact domains that PD-1 has, that T cell checkpoint inhibitor. They have a signaling machinery that turns off immune cells the exact same way that PD-1 can turn off immune cells in the T cell. But these Siglec receptors are expressed on a far more diverse collection of immune cells. So PD-1 is primarily on T cells. But there's lots of other immune cell types. There's macrophages, NK cells, dendritic cells, other kinds of cells that are derived from what we call the myeloid lineage. And the Siglec receptors are expressed on all of those other types of immune cells. So it seemed to us that the Siglecs could be a much broader mechanism to turn off the immune system than the T cell receptor PD-1. So we had a hypothesis. Just as the T cell uses PD-1 to engage PD-L1 on the cancer cell, and the cancer cell can exploit this interaction to shut down the T cell, we thought maybe the reason so many cancers overproduce cyaloglycans is because those cyaloglycans can bind to the Siglec receptor family on a variety of immune cells and shut down those immune cells just like the T cell checkpoint receptor. And if that's true, then blocking the interaction between Siglecs and their cyaloglycans could be another avenue to activate the immune system. And maybe that's the missing part in all these patients that don't get a benefit from the PD-1, PD-L1 inhibitors. So we set out to test that hypothesis experimentally. And this now is published work from 2014, so I won't go through the gory details, but it was a very important study in our lab because it sort of established a model for how the Siglecs could be exploited by cancers to evade immune destruction. And we focused on a class of immune cells called NK cells. Those are natural killer cells. They have an activating receptor called the Fc gamma R3. And that receptor can form a synapse with cancer cells through the intermediate participation of antibody molecules. And these antibodies are produced by our immune system against molecules on the surface of cancer cells. We call those cancer antigens. Now, when our immune system makes these antibodies and they bind to the surface of the cancer cell, they will allow the NK cell to dock, just like the lunar module docking on the moon, right? So these NK cells will dock. And that kicks off the activating signal. And when that happens, the NK cell will kill the cancer cell. And this is a process that's called antibody-dependent cell cytotoxicity, or ADCC in the jargon of the immunologist. So we speculated that if the cancer cell has acquired the ability to overproduce the cyaloglycans, that cancer cell should also be able to engage a Siglec on the NK cell. And for NK cells, that Siglec is called Siglec-7. And so we tested whether that engagement of Siglec-7 could override the activating signal through Fc gamma R3. And it did. And what we learned from that is not only are these cyaloglycans able to confuse the NK cell and protect the cancer cell, they can also give that patient resistance against some important cancer medicines. For example, there's a breast cancer medicine called Herceptin that Genentech and Roche produce. And that drug is an antibody that binds to a breast cancer antigen. And when a patient responds to that drug, it's because their immune system, their NK cells, can come and kill the cancer cell when the antibody is there. But many patients are resistant to this drug, and nobody knew why. And what we found is that the cyaloglycans are making patients unresponsive to the drug because of their engagement of Siglecs. And same for a leukemia drug called Rituxan. It's another antibody that binds a leukemia cell surface antigen. So after we published that paper, over the next 5 years, there was a slew of important papers from other labs. For example, the lab of Stefan von Gunten, who found that in human patients, tumors were expressing cyaloglycans that bind Siglec-7 and Siglec-9. There was a paper from my own colleague at Stanford, Irv Weissman, who found that Siglec-10 on macrophages can be engaged by another type of cyaloglycoprotein to prevent tumor immune destruction. papers from Heinz Lauble's lab at University Hospital in Basel, from Ajit Farki's lab at UC San Diego, Joy Burchell in the UK, Liping Chen at Yale. And finally, in 2019, there was a very nice overview article in Chemical and Engineering News, which is the American Chemical Society magazine, summarizing all these discoveries as fueling a new breed of cancer immune therapies. So it's the development of those immune therapies that is now the current focus of my lab at Stanford. And once we understood how important the cancer cyaloglycans and the Cyaloglycan receptors are in immune suppression by cancers, it became clear that if one blocks that interaction, one might be able to potentiate the immune system against cancer in a different way from those T-cell checkpoint receptors. So how would you go about doing that? And there were two strategies that we contemplated. On the one hand, you could imagine making medicines that are very much like the Merck drug Keytruda, a monoclonal antibody that binds to the Cyaloglycan and blocks it so it can no longer engage those cyaloglycans on the cancer. So this would be very analogous to those T-cell checkpoint inhibitors that were recognized with the Nobel Prize. But there's a problem with this approach, as we saw it, because there are many Cyaloglycan receptors. There's 14 of them in humans, and at least 9 of those have the right signaling machinery to turn off the immune cell. So which one or ones do you need to block, right? You would have to know for a particular patient, for a particular tumor, whether Cyaloglycan 7 was most important, Cyaloglycan 8, Cyaloglycan 9, Cyaloglycan 10, and so on, or maybe you need to block multiple Cyaloglycans to get a potent immune activating consequence. And that was really unknown, whether some Cyaloglycans were more important than others in a different tumor setting. So without knowing which Cyaloglycans you need to block, it's very difficult to create a therapeutic strategy. So we thought we might take a different approach. Instead of targeting the Cyaloglycans with inhibitors, we said, let's go after their ligands on the tumor cell. Because one thing all the Cyaloglycans have in common is they have to bind to a glycan that has sialic acid as that terminal component. That capping structure is always a sialic acid. So if you can disrupt the sialic acids on the cancer cell, you would basically prevent all the Cyaloglycans from engaging with that cell. It would be a Cyaloglycan agnostic approach to targeting this pathway. So we had the idea of a therapy or a therapeutic molecule that would function like a molecular lawnmower. Imagine a medicine that, like a lawnmower, will park on the surface of the cancer cell and turn on and literally mow the sialic acids off, and thereby deprive that cancer cell from the ability to bind any receptor from the Cyaloglycan family. And so the form that molecular lawnmower took was a conjugate between a tumor-targeting antibody and an enzyme called a cyalidase. And cyalidase enzymes are found in many different organisms, including humans. And these are enzymes that catalyze the hydrolysis of the sialic acid sugar from the stuff that lies underneath it. Okay, so it literally cuts sialic acids off like a pair of scissors. So former postdoc Han Shao, who's now on the faculty at Rice University in Texas, and Melissa Gray, who's a grad student who just defended her PhD about a month ago, they came up with this design. And as a model antibody, they chose the Genentech's drug trastuzumab, which is the active ingredient in Herceptin. It's a breast cancer drug. And trastuzumab binds to a tumor antigen called HER2. It's a protein that's overexpressed on many cancers. And what they did is fused near the C terminus of the heavy chain of this monoclonal antibody, the cyalidase from a bacterium called Salmonella. So this was just kind of a tool molecule to test the principle of targeting a cyalidase to the surface of a cell. And I won't go through the gory details here because this is also published work, but we actually made some mutations to the cyalidase to deactivate it. So it's a very inefficient cyalidase, and it requires the antibody targeting to position it close to the sialic acids that it can then cut. And that was very important in making a molecule that would selectively mow the sialic acids off the cancer cell and leave the healthy cells alone with their sialic acids intact to prevent against autoimmune reactivity. So how did we build this fusion between two proteins, the enzyme and the antibody? Well, we did that with this brand of chemistry that Stefan mentioned when he introduced me so kindly, called Bio-Orthogonal Chemistry. And as Stefan mentioned, this is a type of chemistry that we originally invented to do reactions in living systems, in cells, in animals, and now actually people are doing these chemistries in humans. There's some interesting drug delivery applications. But in this setting, it was also a very useful type of chemistry to take two big protein molecules and chemically link them together in a very well-defined way. And so for the chemists in the audience here, what we did is we used a technique we call the aldehyde tag technique, something else we had developed years earlier in the lab, which allowed us to engineer the presentation of aldehyde functional groups site-specifically near the C-terminus of that monoclonal antibodies heavy chain. And those aldehydes were then substrates for a reaction with this dimethyl hydrazine functionalized indole, which with the aldehyde would undergo a sort of Pictet-Spengler type reaction to form this carbon-carbon bonded adduct. And you can see there's an ethylene glycol spacer here and an azide group at the end. So this chemistry allowed us to basically put a linker and an azide site-specifically where we ultimately wanted to conjugate on the enzyme. And people who are familiar with bioorthogonal chemistry will recognize that an azide is a wonderful functional group to click other reagents to. And so in parallel, we took the Cyalidase enzyme and did chemistry on its cysteine residue to introduce another little spacer terminated with this dibenzo cyclooctine. And the cyclooctine is the partner of the azide. So these two will get together and click together, it's the parlance of this field, to form this, this new adduct where there's now a Cyalidase enzyme on each heavy chain linked through this organic chemistry linker. So having made these reagents, the next question was, are they good at cutting the sialic acids off of the cancer cells that the antibody can bind to while sparing other cells that the antibody can't bind to? So to test this, what Melissa did is she mixed together in the same tube cancer cells that express HER2. That's the antigen for trastuzumab, the antibody. And she mixed those together with other cells that do not express HER2, the HER2 negative cells. And both of the cell types had roughly the same amount of sialic acid on their cell surface glycans. And one can analyze these cells using an instrument called a flow cytometer. And the data for the flow cytometer look like these two-dimensional histograms. So on the x-axis is the amount of HER2 on these cells. And you can see there's two populations. There's this group of cells that are HER2 positive. This other group of cells are HER2 negative. And on the y-axis, we can measure how much sialic acid is on the two cell types. And you can see they're both similar in their density of sialic acids. And we measure that by binding to these cells a protein called SNA. It's a lectin derived from a plant, and it has a fluorescent probe on it. So these cells are fluorescent, and that's what we're measuring. So what happens when you treat this mixed population of cells with what we call T-Sia? That's the trastuzumab Cyalidase conjugate. Well, at different doses of that conjugate, so sub-nanomolar doses all the way up to triple-digit nanomolar doses, you can see that the HER2 positive cells have been stripped of their sialic acids with no reactivity on the HER2 negative cells until we get to the very highest dose. So even at this sub-nanomolar dose of that enzyme conjugate, the HER2 positive cells have dropped out of the top quadrant and into this lower quadrant. Their sialic acids have been removed. And as you dose out, you can see they're just completely, totally removed, even at 6 nanomolar dosing. And by contrast, those HER2 negative cells in this upper left-hand quadrant are essentially unaffected by this reagent until the very highest dose. They start to drift a little bit down. But this is good because it means there's excellent on-target reactivity of this conjugate with minimal off-target reactivity. And that experiment, combined with some other cell culture experiments we did, gave us some confidence that we could take these reagents and test them in a mouse model of HER2-positive breast cancer. And toward that end, we formed a collaboration with Professor Heinz Laubli, who's at University Hospital in Basel, and his graduate student, Michael Stanzak, was running this model called the EMT6 HER2 breast cancer model. So basically, this is a model in which mouse breast cancer cells are engineered to express human HER2, which is what's on the human breast cancer. Then these cancer cells are injected into the mammary fat pad of these animals, and tumors grow just like a breast cancer would grow. And once those tumors had reached a certain volume, Michael injected into the tail vein of these animals either just a saline solution, the naked antibody trastuzumab, that's the human drug, the clinically approved drug, or the trastuzumab Cyalidase conjugates at 2 different doses. And what he found is that the naked antibody trastuzumab is ineffective in reducing the progression of disease in these animals. This is a trastuzumab-resistant breast cancer model, and that was already known in the literature. But when there's a Cyalidase attached to the antibody, now you have an effective cancer drug that really limits the growth and the progression of this very aggressive tumor in this animal model. So data like these then motivated us to think about how would we translate this concept into a human therapeutic. And a great way to do that is to form a company and to finance that company in a way that allows that clinical translation. So together with my colleague, Paul Crocker, who's an immunologist at University of Dundee, and entrepreneur Jim Broderick, who had a track record of successful startup companies, the 3 of us formed a company called Paleon Pharmaceuticals, which is physically located out in the Boston area. It's a wonderful location for companies in the cancer space and kind of equidistant between myself and Paul Crocker, between California and the UK. And the brilliant scientists at Paleon have spent the last several years making a developable human therapeutic candidate on this concept of the antibody enzyme fusion. And they made some very important improvements from our initial proof of concept design. First and foremost, they swapped out this bacterial enzyme, which would be immunogenic in a human probably, for one of the human sialidases called NU2. And then the other change that they made is rather than stitching the enzyme and the antibody together chemically, they created a construct in which there's a genetic fusion between the enzyme and a component of the antibody. And they also made the whole thing a bit smaller by simply swapping out one of the FAB domains of the antibody for the enzyme, which is similar in size. So this is what their molecule looks like. And they're right now in the process of manufacturing this molecule to start a phase 1 clinical study in the first quarter of 2022. So hopefully we're just a few months away of testing this concept in humans. And we hope that we have a potent immune therapy, which more patients can respond to. So that's how that part of the project is unfolding. But we still have a lot to learn at the level of the basic science. And one question that has been lingering in our minds and everybody's mind is, we know observationally that cancer cells often overproduce these cyaloglycans. But what really wasn't clear is how that happens inside the cell, right? Something has changed in the cancer cell that has allowed it to overproduce all these cyaloglycans. And if we could understand the pathways inside the cell that lead to this tropical rainforest on the cell surface, maybe we could find some other targets for immune therapy, and maybe even with small molecule drugs rather than these biologic drugs. So how does this happen? And we had a clue to how this might happen from a publication that came from Stanford just around the time that I was moving there from UC Berkeley. And that observation came from the lab of Dean Felsher, who's an oncologist who studies the transcription factor MYC. Now, MYC is a very famous oncogene in the cancer field. MYC is overexpressed in a majority of human cancers. And as a transcription factor, what it does is it turns on the expression of hundreds of genes that are important in cell growth and proliferation. So MYC overexpression can drive the cell to grow and grow and grow. What wasn't known until Dean's publication is that MYC can also drive the expression of genes that allow the cancer cell to suppress the immune system. And in 2016, he published a paper in Science showing that MYC can lead to the overproduction of PD-L1, which is the ligand for the T-cell checkpoint receptor PD-1. Okay, and here's that paper here. MYC regulates the anti-tumor immune response through another molecule, CD47, but also PD-L1. And when we saw this publication, we couldn't help but wonder, given the analogy between how PD-1 suppresses the T cell and how the Cyaloglycans suppress many immune cells through that same signaling biochemistry, could it be that MYC is also driving the upregulation of those cyaloglycan ligands that engage with the Cyaloglycans? So we wanted to test that idea experimentally. We formed a collaboration with Dean Felsher's lab, and in particular with his postdoctoral fellow, Anya Deutzmann. And Anya had these beautiful transgenic systems that she worked with, both transgenic animals and transgenic cancer cell lines, in which she had engineered the expression of MYC to be controllable with a small molecule switch. And the small molecule is an antibiotic called doxycycline. And so the way that her model works is she can take the cancer cells, and in this case, it was a T cell acute lymphoblastic leukemia cell line. And by adding doxycycline to the cell culture medium where these cells are growing, she can basically shut off MYC. And when she takes away doxycycline, that turns on MYC. So these cells are otherwise identical. It's just plus or minus the expression of this oncogene. And then once you have that switchable system, you can ask all these interesting questions like, when MYC is on, what other genes get expressed that are controlled by MYC? And in our lab, we said, well, we can also use mass spectrometry to look for changes in the structures of the glycans when MYC is on versus MYC is off. And furthermore, we had Cyaloglycan reagents that were soluble Cyaloglycans fused to the Fc region of an antibody that we could use as probes. When they bind to the cell, that would tell us that there are ligands for Cyaloglycans, sort of functional ligands that could be controlled by MYC. So Benji Smith, who was an MD PhD student in my lab, did these experiments with the MYC on and off system. And what he found was quite striking. So this is a volcano plot, as we call it, that looks for changes in the transcription of certain genes. And these red dots are genes that are cranked up when MYC is on, and the blue dots are genes that are turned down when MYC is on. But the most important finding from all of this was this one gene up here, this outlier red dot. This is a transcript that is driven by MYC. So when MYC is on, this gene gets turned on. And it's a gene that encodes an enzyme that is a sialyltransferase. That's an enzyme that builds cyaloglycans. And specifically what ST6 GalNAc4 does is it takes a trisaccharide structure called the sialyl T antigen, and that's just historic. And this structure has a pink diamond that's a sialic acid linked to two other sugars. And this enzyme will add another sialic acid to make a disiallo structure called the disialyl T structure, starting from this monosialyl structure. And we confirmed that this was an important change mediated by MYC expression with our mass spectrometry study of the glycans on these cells. So for example, when MYC is on, that disialyl T structure shows up in the mass spectrum as a dominant structure on the cell surface. It is the most abundant form of what's called the O glycans. And when MYC is off, the abundance of that structure drops dramatically, almost in half. So there's clearly a switch that's sort of turning up that disialyl T structure or turning it down. And so some more experiments led to this model that we proposed, in which when the MYC oncogene is on, that cranks up the action of ST6 GalNAc4, that sialyltransferase. So here's MYC sitting on the genomic DNA, driving the transcription of genes. ST6 GalNAc4 is one of those genes. Now there's an increase in the production of this disialyl T structure, which ends up populating the cell surface. And this monosialyl structure goes to the disialyl structure. There's more sialic acid, and that particular structure binds with high affinity to Cyaloglycan 7, which is on a lot of different immune cells. And that can then block the activity of the immune cell. So this model suggests that if you take away ST6 GalNAc4, you should be able to deprive the cancer cell of the ability to make this structure and then deprive it of the ability to shut down the immune system. And we tested that genetically by knocking out ST6 GalNAc4 using CRISPR technologies. So this is the work that Anya did. She took this leukemia cell line and just broke up the gene encoding ST6 GalNAc4. And then she took those knockout cells and tried to grow tumors in a mouse. And what she found is that those tumors do not grow well once they've lost this enzyme. And the reason is that the immune system is able to infiltrate the tumor and kill the cells as they're trying to proliferate. Whereas normal cells that have that enzyme, they just grow and grow and grow. Those tumors get bigger and bigger because they're tricking the immune system through the expression of that diCyLLT structure. And the implication of this is that if you had a drug that bound to ST6 GalNAc4 inside the cell and blocked its catalytic activity, that molecule could be an immune therapy. So this is now a new direction that we're taking in the lab to try to discover small molecule inhibitors of the sialyltransferase ST6 GalNAc4. So hopefully all of this has taught you something about the important role of cell surface glycans in the immune system, and in particular in cancer. And hopefully we'll be able to deliver to patients a new class of drugs. So with that, let me thank all the people whose work I mentioned. I called people out by name and by face, but there's a supporting cast, of course, which is every lab member has something to contribute. And then none of this would be possible without the generous funding from these various agencies, the NIH and the Howard Hughes Medical Institute. And with that, I'll thank again the people who invited me and who've organized this seminar series, and open the floor up for questions. Thank you. Okay, now I think the questions are going to either come, I don't know if they're going to come in the chat or if they're going to come in the Q&A. I'm not sure. I do see a hand up, Dr. Zeilinger. Is there a question that is associated with the hand?
Okay. I was actually struck by the immense progress which was made in your field and by you over the recent time. And one of the remarks you made was that this sugar coating was discovered by, as you said, a primitive technology about 70 or 100 years ago, right? So this standard microscope. Well, at that time, this technology was not primitive. My question simply is, what are your wishes for a technology in 20, 50, or 100 years?
Oh, that's just such a great question.
What would you like to have?
Right now, there's a big disconnect between what we can study at the level of the cells and tissues and the molecular information of the structures of the glycans. So the only way that we can get that actual chemical atomic structural detail is through mass spectrometry. But we have to grind up and destroy the tissue and break apart the molecules in order to get the chemical structure. Right. So for example, that diCyLLT structure, we figured that out by mass spectrometry, but we can't get that information from the intact tissues and cells. So that means we can never really know what's the spatial distribution of that structure, right? That's a huge gap. So in my fantasy, and hopefully it won't take all the way till 2050, hopefully it's on a shorter time scale, it would be wonderful to have a technology in which we directly analyze the tumor tissue the biopsy from the patient, and see what are the molecular structures of the glycans in the specific places. Yeah, we don't have that, right?
Great. Okay, again, thank you for a fantastic talk. I learned a lot, even as I'm from a completely different field. Thank you.
Thank you. So now I see some questions are starting to appear in the Q&A box. So that will be now the site for Q&A. And the first question I'm seeing here is, what if any effect do cell surface glycans have on abnormal cell adhesions in cancer and other diseases such as fibrosis? And there's actually quite a body of literature that focuses on cell adhesion and in particular metastasis. So the process of metastasis involves cancer cells breaking free of a primary tumor. So they're de-adhesion, right? The opposite of adhesion, they're separating. They travel through the bloodstream and then they get out of the bloodstream into a remote tissue site, take up residence there and start to proliferate there. That's a metastasis. And so the leaving of the solid tumor and the engaging of a new tissue environment both involve some complex adhesion processes. And there is literature showing a role for different kinds of glycans in that process. And in particular, there's a family of homing receptors called the selectins. They're separate from the Siglecs, just a different family, that are known to allow immune cells to get out of the bloodstream and get into tissue sites. And there's some work that shows that circulating cancer cells can actually hitch a ride on your immune cells to help get themselves out of the blood and into a remote site for metastasis. So that could be a very important component of cancer. People study that. Fibrosis, I'm not so sure. That's just a condition I don't know as much about, but macrophages are thought to play an important role in fibrosis. And macrophages, they get around the body by these selectins and glycan binding receptors. So there could be something there as well. It's a good question. Okay, next question. Thank you for the great talk. You're welcome. I was curious how much is known about the mediation of the interactions between platelets and cancer cells in the context of metastasis by cyaloglycans. And again, this really touches upon what we were just discussing. Platelets, when they're activated, express one of the selectins. It's a selectin called P-selectin because it was originally found on platelets. And again, there's some work suggesting that P-selectin can allow those platelets both to aggregate with other immune cells in circulation, but also to attach themselves to the endothelial surface where there are glycan ligands that can bind to P-selectin. And so to the extent that circulating cancer cells might form clusters or aggregates with platelets, they might be able to hitch a ride on those platelets to get themselves to a distant site. And again, there's some beautiful work from Ajit Varki's lab at UC San Diego that points to that selectin interaction as important in metastasis. Okay, the next question. Do you think that therapeutic approaches using this chemistry are likely to have generic toxicity issues due to interference with normal cellular processes? We definitely worry about the off-target consequences of having a cyalidase floating around in the human body in the plasma. The biggest risk, as we see it, is that if that enzyme does take sialic acids off of some normal healthy cells, there's a risk of allowing immune cells to react against healthy cells and have an autoimmune reaction. That could be an acute autoimmune condition like a cytokine storm, or it could be a chronic autoimmune consequence like the development of type 1 diabetes or rheumatoid arthritis or Crohn's disease. These are all chronic inflammatory conditions. And the reason I think that is a legitimate risk is because those are exactly the off-target consequences of the T-cell checkpoint inhibitors, the PD-1 blockers. So patients respond to those drugs with an anti-tumor immune response, but at the same time, they do have some autoimmunity. They can have fevers and hemorrhagic reactions. Some of them end up in the emergency room. So those immune therapies are, there can be some dire consequences. It's a risk that a patient will take when they're out of other options. And those immune therapies have actually cured people of otherwise fatal cancer diagnoses. Like the most notable example is probably Jimmy Carter. Okay, he's a former president of the US. He was in his 90s, and he had melanoma that had metastasized to his brain. And he was one of the first patients to be treated with Keytruda, the PD-1 blocking drug. And he was in the lucky 20% or so that actually was cured of his disease. It's quite spectacular. But again, the side effects can be brutal. But a patient is, willing to sign up for becoming a type 1 diabetic rather than die of melanoma in the brain, right? So it's a trade-off that people will make. So we're going to watch out for that. Next question. Is there any link between the Cyaloglycans and cell metabolism, like glycolysis and the Warburg effect? Great question. And any link with aging and Cyaloglycan biology? Two outstanding questions. There is a potential connection, actually, between sugar metabolism for energy, as we think of it, and cell surface glycosylation patterns. There is a potential connection there. So the Warburg effect that this person is referring to is an observation that many cancer cells become addicted to sugar as an energy source. They require glucose, as opposed to a normal situation where cells can actually use fat as a primary energy source. But some cancers actually, they lose the ability to use fat as the primary energy source, and they really become addicted to sugar. And glycolysis is the process by which cells can take glucose and break it down and get energy out of it. So it turns out that glucose can also go through a pathway called hexosamine biosynthesis, which leads to another intermediate, which is a critical intermediate for sialic acid biosynthesis. So you can actually chart a pathway by which glucose goes to another intermediate called UDP N-acetylglucosamine, which then goes to sialic acid. So it's possible that having a lot of glucose around can actually drive sialic acid biosynthesis just through metabolic flux. But there hasn't really been a good connection made between the Warburg effect and the sialylation status of a cell experimentally, but theoretically, there's possibly a connection there. So I'd say the jury's out, but it's not outlandish to actually do the experiment. Aging is another interesting question. There's actually evidence that Cyaloglycans change in their expression levels in the brain during aging, at least in mice. And this is potentially important for a class of immune cells called microglia. So microglia are like macrophages in the brain, and macrophages are immune cells that gobble stuff up. They're hungry and they eat things and they get rid of garbage. So the microglia in the brain are thought to play a role in cleaning up the brain from protein garbage. And this observation is that when people age, they actually have an increase in Cyaloglycan expression on the microglia, which makes the microglia less active, less hungry, less able to clean the garbage out in the brain. And this could be important for a number of neurodegenerative diseases for which a hallmark is the accumulation of protein garbage. Alzheimer's disease, Parkinson's disease, ALS. These are all neurodegenerative conditions that are marked by the accumulation of garbage protein. So it could be that having dysfunction of the microglia contributes to those diseases. And if you block microglial Cyaloglycans and make the microglia more active and more hungry, that might be a therapeutic strategy. That's been modeled in the mouse. And in particular, there's a colleague of mine at Stanford. He's a neuroscientist named Tony Weiss-Corey. And Tony and I have actually published a few papers in this space, thinking about the Cyaloglycans as targets for neurodegeneration. So I would encourage you to look up those papers, or I can share them offline for anyone who's interested. Next question. Tumor environment also has certain rare sialic acids, like this, a structure called free KDN, which is an analog of sialic acid in which there's been a loss of an acetaminophen group. Can we utilize such structures to develop therapeutics to arrest cancer growth? You know, Ajit Varki, again, at UC San Diego, is a leader in this thought process about chemically altered sialic acids as natural tumor antigens. And his lab, as well as a company that he co-founded, is making antibodies that are specific for these rare forms of sialic acids in order to, potentiate NK cells and macrophages against cancers. So whether that is a viable strategy for human therapeutics has, I think, yet to be determined, but he's published some work in animal models that looks promising. So I would definitely keep your eye on Ajit, as well as his company. I think the company is called Cyamab, like a sialic acid monoclonal antibody, and they're located in Boston. So I think we're going to find out if this is a good strategy, but it's certainly a good idea. Another question. I am curious to know if these orthogonal chemistry techniques can be applied to block microglial responses in neurodegenerative diseases. There it is. Especially your comment in crossing the blood-brain barrier. Okay, so I just talked about that, microglial Siglecs and neurodegeneration, so that's timely. The blood-brain barrier obviously is a problem for biologics. So monoclonal antibodies against the checkpoint inhibitors or the Siglecs, however you develop them, they don't get across the blood-brain barrier very effectively, very low exposure there. There are companies that are trying to improve upon that with so-called brain shuttle attachments. Denali Therapeutics in South San Francisco is kind of a leader in this space. But generally speaking, you have a much better shot at high brain exposure with a small molecule than with a big biologic. So I think if somebody wanted to take on the microglial Siglecs and block them to increase their activity in the brain, a small molecule inhibitor might be a good way to go. Siglecs are not the worst targets for small molecule inhibitors. They're definitely challenging by nature, but I think that there's a way to approach that problem. And people are probably working on this. I know there's a number of companies that are going after Siglecs for neurodegeneration, and they might have small molecule programs. Next question. Cyaloglycoconjugates can differ between mouse and human. Absolutely true. In addition, the Siglec family is rather fast evolving and also differs in part significantly between mouse and human. Yet mice are one of the most important model systems to study cancer immunology. What do we do about this problem? Okay, yeah, so the mouse, you're right, is not the best model for Siglec biology, but it's really the only game in town right now. And it is a big frustration for people in this field. So for example, we focused on Siglec 9 and Siglec 7 in our lab because those are the Siglecs in humans where you find them on immune cells in the tumor microenvironment. But the mouse does not actually have a direct ortholog of Siglec 9 or of Siglec 7. It has something else, which is kind of in between the two, called Siglec E. So, we use the mouse model and we treat Siglec E as if it's some hybrid between Siglec 7 and Siglec 9, but we know that that's a very imperfect model. So what are people doing about this? Well, first of all, there's some immunologists that have decided that mice are useless as a model for any human immunology, and they've thrown them all away and are trying to find other ways to use human subjects and human blood samples to test their hypotheses. Mark Davis at Stanford is an example of a person who's just given up on mice altogether. For cancer, though, I think you really do need animal models like the mouse. So Jeff Ravitch at Rockefeller University is taking the approach of basically making transgenic mice that are engineered to express human Siglecs in the right immune cells that you would find them in humans. And he published recently on a transgenic mouse that expresses both human Siglec 7 and human Siglec 9 on the right myeloid immune cells and activated T cells where you would find them in humans. So that's one step towards humanizing a mouse to have Siglecs that are more like the human Siglecs. But those mice still have the mouse Cyaloglycans, right? That could be different from humans. So we're not there yet, but yeah, I will just agree that it's a problem and a frustration. I think the key is to get your therapeutic candidate into human subjects as quickly as possible. Next question. What do you think about terminal sugar residues like fucose having some role in cancer? Yeah, so there's a fucose story brewing out there. Observationally, people have found that certain fucosylated glycans are often overproduced in cancers compared to healthy tissue. So, there's this observational story that's looking like it's quite parallel to the sialic acid story. But so far, to my mind, what's missing from the fucosylation relationship with cancer is mechanistic insight. So, back in the 1960s and 70s and 80s, people were finding the cyaloglycans overproduced in cancer, but there wasn't any idea as to why, what the significance of that was. And one couldn't really have a hypothesis until the Siglecs were discovered. And most of them came out of the human genome sequence, right? A few of them were known before that, but not all of them. And once you saw how many Siglecs there were and what immune cells they were on, you could then speculate, oh, maybe this is why the cyaloglycans are produced on the cancer cell. That's a phenotype that arises through natural selection because those cells can escape the immune system. With the fucosylated glycans, we don't yet have that mechanistic insight as to what natural selection process might drive their overproduction. And so I think the field is sort of waiting for that connection to be made between the fucosylated glycans and some biology that's important in cancer. And there's some speculation out there. Some people will draw upon the selectin family of receptors, which do bind to fucosylated sialylated glycans as potential players. Actually, some of the Siglecs seem to have a preference for sialoglycans that also have fucose. So there could be an intersection between the fucosylation phenotype and the Siglec biology. But it really hasn't been, I think clarity is still missing from that. And so there's work to do there. Next question. Sialoglycans have Siglec binding partners, making them great targets. Do you see a similar application to target other types of glycans? Oh, it's back to the fucose. Okay, sorry, I didn't see where that was going. Yeah, I think we basically covered that. And I think it would be great if we had more clarity on why fucosylated glycans are sometimes overproduced in cancer. And that would then focus us on new targets. And then finally, can we do differential sialoglycoproteomics in cancer stem cells? And if yes, what's the right control? Oh, this is a great question. I know people certainly are interested in doing molecular characterization of cancer stem cells to see what differentiates them from other cells, because the principle which Stemcentrx went after, and they were acquired by AbbVie, the principle is if you could get the cancer stem cell, then you've cured the patient. And until you get the cancer stem cell, there's always the risk of recurrence. So people have done proteomics on cancer stem cells, but not an extensive amount of glycoproteomics or glycomics. And I think one of the challenges is it's very difficult to isolate an abundance of cancer stem cells so that you have enough material to do the kinds of mass spec that's required for glycoproteomics, which is much more difficult than just for kind of straight up proteomics. So people have definitely found markers on cancer stem cells that are heavily glycosylated proteins. For example, certain mucin glycoproteins are found on cancer stem cells, and they are heavily glycosylated and could be carrying sialoglycans that are relevant for immune modulation. But again, without being able to purify with abundance, those cells to do the molecular characterization by mass spec, it's really difficult to ascribe to them a unique glycosignature that you might not find on the control. What is the right control? Good question. I'm not the right person to answer that, but presumably all the progeny of the cancer stem cell that have differentiated, into what ultimately is the tumor. Okay. And that's the end of the Q&A questions. So unless there are other lingering questions, maybe it's a wrap.
Thank you very much again for this outstanding talk. And as Anton Zeilinger said, also non-biochemists could take away a lot. Of course, also myself. And so with this, I'm handing back to Tom Hensinger for a few concluding words.
Okay, also from my side, thank you. Thank you very much for this fantastic talk, also on behalf of the audience. And I also like to thank everybody in the audience for joining us today. And we hope to see you again soon at any one of our events. And I invite you to visit our websites, IST Austria, to find out more about our events and activities. And it remains that I wish you a very pleasant evening and a safe and a healthy time during these difficult weeks. Thank you, everybody.
Thank you. And if any Americans in the audience, happy Thanksgiving.